Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Preliminary results from an ongoing trial by Intellia Therapeutics show that a CRISPR-Cas9-based drug can be delivered into the body to target the liver and reduce expression of the gene that causes transthyretin amyloidosis, ATTR. This is the first clinical trial demonstrating successful in vivo. Some previously approved drugs accomplish this with small interfering RNAs or antisense oligonucleotides that help degrade or sequester the RNA encoding TTR. 2, 3 But these treatments require a pre-treatment followed by weekly or monthly administration of the drug, which causes unpleasant side effects. These treatments often fall short of curing the disease. While symptoms improve, many patients die within 10 years of diagnosis. ATTR can lead to the accumulation of TTR protein in various organs, especially the heart, shown in yellow. The goal for Intellia is not only to halt progression of the disease, but potentially to be able to revert and gain function, said Sepp-Lorenzino. Using CRISPR to edit cells directly in the body, rather than removing and editing cells in the lab and returning them to the patient, is no small task. Wrapping a drug in a bubble of fats called a lipid nanoparticle causes it to head straight to the liver, enabling efficient delivery and reducing off-target effects. Intellia's lipid nanoparticles contain a guide RNA targeting TTR and mRNA encoding the Cas9 endonuclease that makes the genomic cuts. In their ongoing Phase 1 trial, they administered the drug, Intellia 2001, to 6 patients with nerve damage resulting from ATTR. Half of the participants received a lower dose and half received a higher dose. 28 days after treatment, there were no reports of severe adverse effects. For comparison, existing therapies reduce its presence by roughly 80%. He was surprised to see that some patients in the trial reached these desired levels already. It opens up the real possibility that these patients are actually going to improve, Gilmore said. Future plans are to continue increasing the dosage, which Gilmore expects will lead to better outcomes for more patients. There are plans to test the drug in these patients in the next phase of trials. It's going to shift from a model where we're taking pills every day or injections every few weeks for long periods of time, Musunuru said. Gene editing therapies are going to transform that model into one where you have one-and-done therapies. Demonstrating the ability to deliver genome editing machinery to the liver potentially opens the door to editing any gene in the liver and to treating more conditions. For example, scientists may engineer lipid nanoparticles with specific molecules on their surface to interact with a target cell type. An in vivo clinical trial conducted by Adidas, a biotechnology company designing CRISPR-based therapeutics, to treat a retinal degeneration disorder with virus-delivered CRISPR-Cas9 also recently showed clinical benefit, although retinal editing doesn't have as clear of a readout as TTR levels. I think that's the real challenge for the wider application of this technology. References J.D. Gilmore et al. CRISPR-Cas9 in vivo gene editing for transthyretin amyloidosis. N. Engel J. Med. 385. 6, 493-502, 2021. D. Adams et al. Patisiran. An RNAi therapeutic for hereditary transthyretin amyloidosis. N. Engel J. Med. 379. 1, 11-21, 2021. M.D. Benson et al. Inoderson treatment for patients with hereditary transthyretin amyloidosis. N. Engel J. Med. 379. 1, 22-31, 2021. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.